क्या है। सो विद इन अ सेकेंड आई थिंक सो हम लोग होने लग जाएंगे। थोड़ा सा आप लोग चेक करो, एनीवन, उर्वशी और श्रुतिका, है ना?
जी मैम, चेक करिए।
आप लोग चेक करो, एनीवन, उर्वशी आने लग गया है।
हाँ मैम, आने लग गया, आवाज आ
ठीक
रही
है,
है।
बढ़िया है, सब बढ़िया दिख रहा है।
हाँ।
गुड मॉर्निंग नीतू मैडम।
Good morning, ma'am. Good morning, Nitesh sir. Good morning, all.
Good morning, ma'am.
Good morning.
Looking very pretty, ma'am.
थैंक
निहारिका मैम,
यू।
सब अच्छे लग रहे हैं। उर्वशी मैम, श्रुतिका मैम, मुझे जितने दिख रहे हैं।
नहीं, पहले तो मुझको ही बोला मैडम।
गलत बात है मैडम, आपने हमको नहीं बोला।
नहीं नहीं,
नहीं, पहले मुझको बोला श्रुतिका।
सीनियर से स्टार्टिंग होनी चाहिए ना।
नितेश सर आवाज नहीं,
सीनियर्स,
नितेश सर आवाज नहीं आ रही।
हाँ, नितेश सर की आवाज प्रॉपर।
लाइव चल रहा है मैम, ये बोल रहा हूँ मैं।
ओ सॉरी।
आप लोग अपनी अपनी तारीफ बाद में कर लें।
यस।
माय वॉइस इज ऑडिबल नाउ? आ रही है?
यस सर।
ठीक है।
मैम स्लाइड शेयर करो, स्टार्ट।
अंकुर शर्मा सर को बुला ले मैम, ही इज
नहीं
ऑन
सर।
मीटिंग रूम, तो मैं सोच रहा हूँ बुला लेते हैं उनको, है ना?
9:56 हो गए। अभी तो कोई फ्लोरल वेलकम नहीं करना है ना?
नहीं, जस्ट उनका ऐसे ही हो जाएगा, मतलब वेलकम जैसा वो। उर्वशी मैम,
सर आप ना अपना ये थोड़ा पास में रखिए।
हेलो। हाँ ठीक है।
हाँ, नाउ इट इज वेरी क्लियर।
चलो जस्ट मिनट, मैं फैन भी ऑफ कर देता हूँ, तभी वॉइस क्लियर आएगी।
मैं स्टार्ट कर रही हूँ स्लाइड शेयर।
हाँ, आप स्टार्ट करिए। यूट्यूब पर भी कोई नजर रख ले।
हाँ मैम, मेरा इधर ऑन है।
और उसका वॉइस म्यूट कर दो आप यूट्यूब का।
विजिबल है स्लाइड्स?
हाँ, हमें तो है यहाँ पर।
यूट्यूब पे भी स्टार्ट हो गई।
हाँ, विजिबल है।
I think Dr. Sharma is coming within 2 minutes. I have invited him personally on WhatsApp. So he will join soon. So we will start our session on time.
जी सर।
ओके सर।
Hello.
Hello, Dr. Sharma is here. Okay.
नो सर।
He is joining soon. I think I have already told
ओके
him.
सर।
Mahavi sir is here.
नो सर।
Okay.
Sumit sir is not also here right now.
And I have already shared a link in our group. I think maybe everyone will join soon. So just waiting for Dr. Sharma, then we will start our session.
Okay, sir. Sir, Rathore sir is means.
Rathore sir will come on time, I think so.
No, he is here.
Okay. Rathore sir is here.
I
Very
joined him.
Very good morning, sir. Hello.
Hello, Rathore sir. Hello. Rathore sir is means.
Rathore sir will come on time.
Okay, but he is here.
Okay, no problem. That will be good after completion of Dr. Ankur Sharma lecture. We will start lecture of Dr. Man Singh Rathore, sir.
Yes, sir.
Hello, good morning, Hitesh ji.
Very good morning, sir. Welcome once again.
The program is when it is starting?
Sir, definitely we are starting very soon. So firstly, we have a scientific session of Dr. Ankur Sharma.
Okay.
And after that, we will start your session, sir, in continuation, sir.
Just to check, my voice is audible? Am I audible?
Yes,
Yeah,
it is audible.
it is audible.
It's clear? Okay.
Yeah, yes, sir.
So,
Rathore sir, good morning.
Good morning.
I am Niharika Gokhale, your student.
Yes, I remember your name by the Niharika Subedar. Actually,
Subedar, yes.
Yes. This is one and same name, no?
Yes, sir. After marriage, it is changed to Gokhale.
Yes. The name striked in my mind. Okay,
Thank you so much, sir. You remembered me. My pleasure, totally.
Definitely, Mansoor, Vianata College of Pharmacy memories can never be vanished.
Yes, sir.
All of you are very good students.
Thank you. So Dr. Ankur Sharma is here.
Hi everyone.
Hi, Dr. Sharma.
Hello.
Good morning, sir.
Hello, Mahendra sir. How are you?
Hello, Ankur. कैसे हो?
I'm fine, totally fine. आप कैसे हैं?
बस बढ़िया, फर्स्ट क्लास हैं.
बढ़िया, आप तो बिल्कुल change. Nobody has changed actually. वैसे के वैसे ही हैं.
Thank you. It is a big compliment for me also.
Dr. Sharma is looking gorgeous as always.
Yes, he is.
It's all your best wishes, Nitesh.
Yeah.
So we can say evergreen Dr. Sharma, no?
Yes.
Is this program made online?
Yeah, sir, definitely.
It's online.
Yes, it is live on YouTube, sir.
Okay. That's great. That's good.
Yes, sir.
So I think now we
can start. Yes, sir.
Yeah, sure.
Can I begin, ma'am?
Yes.
A very pleasant morning to all excellencies, respected faculties from various colleges, and dear students. I, Urvashi Sharma, cordially welcome you all on the second day of International Conference entitled Innovation in Pharmaceutical, Medical and Biosciences 2020. Proceeding further, I would like to request Dr. Nitesh Jain sir, Principal UIT, Oriental University, Indore, to give some briefing about the conference and the day. I request you, sir, to please proceed.
Okay, thank you, Urvashi ma'am, for inviting me. So, ladies and gentlemen, first of all, a very good morning to one and all. After completion of first day of International Conference on Innovation in Pharmaceutical, Medical and Biosciences, now we are moving towards our second day. So I will brief what activities generally we will conducted in second day of conference. So there will be a 3 scientific session from our respected all speakers of the conference. And we will also conduct valedictory function after the completion of scientific session. So on behalf of Oriental University Indore, we welcome all the keynote speakers of today's session. So Dr. Ankur Sharma, he will discuss about the role of predictive model in pharmaceutical discovery for clinical scientists, clinical
Translation.
and yeah. So, and we have another scientific session of Dr. Mahen Singh Rathod, sir. He will brief us about current perspective of new drug and clinical trial regulation in India. And the third scientific session will be taken by Dr. Piyush Jain, sir, abdominal pain and irritable bowel syndrome, animal models to clinical studies. So these are the activities which we will do in second day of conference. And at this platform, I really congratulate all my team members for completion of first day of conference. It was really a grand success.
Nitesh sir, your voice is not audible.
You're on mute, Nitesh. Okay.
Hello.
Yes,
So
yes.
on behalf of Oriental University Indore, I welcome all the keynote speakers of today's session. So first speaker is Dr. Ankush Sharma. So I would like to call Dr. Urvashi once again to introduce him for further proceedings of scientific session. Please.
Okay, sir. Thank you, sir. Now, let me to introduce the profile of our first speaker of the day, Dr. Ankur Sharma, sir. Dr. Ankur Sharma, sir, is a senior scientist, PPDM, quantitative pharmacology and pharmacometrics, MRC. Sir has done his graduation from University of Rajasthan, Jaipur, post-graduation from B.R. Nahata College of Pharmacy, Mandsaur, and Ph.D. from College of Pharmacy, University of Oklahoma. Dr. Ankush Sharma sir also has experience of working as a research associate at Pyramid Life Sciences and ORI's fellow intern at FDA. His area of research interest is utilization of PKPD tools for drug development and has also expertise in bioanalytical method development, modeling and simulation, designing experiments utilizing model approach, and protocol writing for IACUC approvals. Once again, we welcome you, sir, and request you to please take the session ahead. But before moving further, I would again request Dr. Nitesh Jain, sir, to please welcome our first eminent speaker, Dr. Ankur Sharma, sir.
So on behalf of Oriental University Indore, it's a privilege to me to welcome my dearest friend, Dr. Ankur Sharma. So welcome, sir, in the scientific session of Icipmbs 2020. Sir, please proceed further.
Okay. Thanks, Dr. Nitesh, and thanks everyone and the coordinating committee and the leadership team for setting up a very nicely, setting up a good grounds for this international conference and providing this opportunity for me to showcase efforts in quantitative pharmacology to the broader audience. So let me share my screen. Okay. Okay. Okay, so can everyone see my screen?
Yes, sir.
Okay, okay, good. So thanks, Urvashi, for the detailed biography so far for me. Um, so my topic for today is the role of predictive modeling in pharmaceutical discovery for clinical translation. So this overall learnings and lessons have been through a myriad of journey through different schools and different stages. So after I graduated from pharmacology, I entered into Pyramid and then College of Medicine, OU, and then did an ORICE fellowship at United States FDA. Followed by that, I joined Merck for learning new approaches in quantitative pharmacology and how modern informed drug development can really pave the direction for drug development and provide the drugs or medicines to patients in a timely manner and cost-effective way. So let me remove. So Nitesh, I see this admittancy waiting hall. How can I remove this part so that I can see my screen?
There are
Okay,
some
okay,
technical
okay,
problem.
okay.
It's not
I think
visible
it's
on.
fine.
Okay, okay.
I think
Okay.
I think it's fine. So as the topic for this conference is like innovation in pharmaceutical industry, so I thought to put up this slide like from where the concept of predictive modeling came into the knowledge. So earlier, this predictive modeling information or the concept was more prominently used in aviation industry. So earlier, the engineers or the scientists, they were always wondering like what should be the right aviation model or the airplane model, which needed a lot of money and time to design and lots of scientific efforts. But they might not get the best prototype of a best robust aeroplane. So what they did, they learned from their from different parameters and different knowledge gaps while doing their research and development, applied into some models and simulation by incorporating their knowledge gaps and parameters by into their statistical models and come up with a more robust design of an aeroplane. And that's why you come with a best prototype of an airplane, which is designed in a way which is more robust, more efficient, and more error proof and safety, of course. So I wish to put a note at the bottom that what is an innovation? Innovation is not only like a new thing comes into market, but also how we can repurpose a science or technology for a broader area. For example, drug development or healthcare industry. So the same concept was further was was initiated by US FDA a decade ago to utilize these model-based drug development approaches to enable or hasten the drug development process and cost-effective way. And this is the paradigm of the different phases of clinical trial. Everyone knows that. But I want to emphasize that it really takes a considerable time the drug reaches from the laboratories to the broader population, and sometimes the drug is biologically is very good, has a good proof of biology and good proof of concept, but because of ineffective trial or a poor drug development, it do not reaches the patient. So the main personality in the entire healthcare industry is the patient. So whatever we do, whatever the development we do for the benefit of the patient. So what we need the right medication, we at an affordable cost and a very effective trial. But sometimes even because of a good proof of biology for the medicines, but the drug trial fails because of a poor trial design or because of an ineffective dose, which so far has been decided more by an educated guess or hit and trial method. So there is an effective, first of all, need for a very effective collaboration between academia and industries for data sharing and due diligence of the sharing of different techniques and tools, so that once the data is available, different approaches can be utilized to get the medicine to the bedside as early as possible and avoid these large delays, high cost, and a higher attrition rate. So that's the overall goal, to get the medicine earlier to the patient at a cost-effective and a more effective way. So I would like to share some of the lessons from an AstraZeneca Big Pharma pipeline analysis. So on the left-hand side, you see a plot which shows which shows different programs at different stages on the x-axis are the different stages in the pipeline, the preclinical, from preclinical to phase 2, and what are the percentage of the programs of the drug development programs were failed. So what we see, and I have highlighted in an oval box over here, that 88% of the programs died at the later stage of the drug development in phase 2 trials, and the major reason for those failures were the efficacy in phase 2B's terminations. Also, I would like to point out that there are 5R framework which governs the successful drug development. These are the medicine should be reaching the right target in the right tissues, and it should be, the drug level in the tissue should be at the safety margin. And of course, there should be like more personalized approach in which you see recently is like a personalized medicine. Personalized medicine in which the right dose, right drug has to be given to the right patient. And of course, it has to have a right commercial potential in like phase 4 clinical studies. On the bottom right, we see that the inability of the drug to reach in an optimum exposure level in the tissues of the patients was one of the key reasons for the lack of clinical efficacy. It means the drug level doesn't reaches the tissues in inappropriate amount, and thereby the proof of biology is not met, or the right exposure, or the PK/PD, which is pharmacokinetics and pharmacodynamics relationship, is not met. So these are one of the key factors which comes from the AZ's AstraZeneca's experience. Now, as a drug development scientist, you always see that you have a good chunk of data from your preclinical research. Does somebody have question? Like I see marks on my screen. Okay, it's gone. So there is always a key question that how I can translate my data from animal studies to enable the proper design for the clinical trials. So of course, you have a chunk of data for the pipeline, but there are a lot of uncertainties in a drug development scientist's mind, or the team, or the industry, that okay, how I can address those valley of death or uncertainties. So in drug development terms, it is called as valley of death, and these uncertainties are basically how we can achieve a robust proof of biology. What should be the right human exposure to modulate the targets? What should be the right human dose, and what should be the right clinical trial design? So you have a good chunk of data available in your preclinical research, which shows like good efficacy and potency of the drug. You also have different physiologies from different species, but how we can assure that you will have a high probability of success in the patients, and how utilizing this data from the animal studies, we can translate that information effectively to have a high probability of success in the clinical trials. So there comes the role of the quantitative modeling and analysis. This science and these tools, they basically enable translation of this preclinical data to a more robust clinical trial design. Okay, and what do we achieve? We achieve a robust proof of biology, a right exposure, a dose, and clinical trial design. So recently, FDA has come up with the Prescription Drug User Fee Act 4th, which is also called as PDUFA Act, which emphasizes on the MIDD, or the model informed drug development, as an effective tool for enabling the drug development or hastening the drug development. It also emphasizes developing scientists for learning different MIDD approaches by different workshops and practices and learning opportunities. So FDA has really set up a ground for developing quantitative clinical pharmacologists for the benefit of patients and society. So you can search on FDA's website about this PDUFA and different policies of the PDUFA. So let's discuss about like what is predictive modeling. So what does this science or tool does is collect the data from the preclinical species, animal studies, the early clinical studies, okay, and try to model those studies and generate the data in a sense that it makes a more refined decision making for the higher clinical trials development. So basically, by doing predictive modeling is like developing mathematical models like a quintessentials of physics and chemistry, like developing different differential equations, just like the physics and chemistry, what we used to do in our early education, and use those mathematical or statistical models to characterize the data and come up with more refined trials and designs. And thereby we can reduce the uncertainties in the prediction of the prediction of any pharmacokinetic response or the pharmacodynamic response. We can mitigate the risks considering the safety and the efficacy part and support the clinical trial designs and doses. So I would like to take this platform as an opportunity to appreciate Louis Shanner. Like he is a person who he's a scientist, Amian scientist, who generated the idea of MIDD approaches to enable utilization or application of pharmacological and statistical models to enable development of the safe drug and efficacious drug, and how it goes by learning and confirming paradigm. Like you learn from the mistakes, you confirm by correcting those mistakes, and then you develop a further clinical trial or further design trial. So I think I took more time on predictive modeling, but let's go on the next slide. So I was thinking of like how I can explain this to the broader audience. So this is one of the good examples, example to explain how an Olympian sportsman uses the stick to cross the bar. So he knows what the bar is, but he uses some tool to cross this bar. So this tool is the threshold which a drug molecule has to cross to become a successful pharmaceutical candidate for patients. And as a drug scientist and drug development scientist, we can utilize these quantitative and translational pharmacological approaches or tools to know that how high the bar is and how much pressure or how much effort I would need from this tool to cross this bar. So just to give a very high level overview to the broader audience. So the commonly applied approaches for the predictive modeling are the bioinformatics tools, which help understand different signaling pathways to understand the downstream signaling, like how the protein regulation would be happening, and eventually how the drug effect would be there in protein translation or in development of, for example, in tumors development. Pharmacometrics, which we more often use, is a combination of applying pharmacology, physiology, and statistical concepts to understand the uncertainties or variability in the responses. That how, if we develop a model for a particular patient, how much we are certain of the fact that different populations, maybe maybe at a different race or different ethnicity or different weight range or different genders, how these different covariates would have a successful drug effect or a successful drug concentration by taking into account the uncertainties. So that's a very, very powerful tool in predictive modeling, which is being widely used. The other one is physiologically based PBPK or pharmacokinetic modeling or PBPK approach, in which apart from the empirical structural modeling, it also takes into account the physiology of the different species, like how the drug basically moves across the different organs in the body, um, and relies on species physiology. The more recent approaches are the machine learning and artificial intelligence, and I would like to emphasize to use these approaches like more cautiously, because because recently I see a more dominant use of these approaches, but these are really high level approaches. These are very complicated approaches, and still these approaches are being explored for drug development in healthcare. So we have to be very cautious about using these approaches, so that the budding scientists and the drug development scientists, they can distinguish between machine learning and artificial intelligence and more commonly used tools for predictive modeling. And definitely it needs a high level of training to understand how we can apply these applications in drug development process. And IBM Watson is one of the good example of how we can how they have done a collective effort of collecting the data from different clinical trials and make them available to the physicians in a more sophisticated way, so that the physicians can learn from those libraries of the data, and can make a judgment on each patient based on each individual history, and very personalized medicine too. So it's very more often it would be coming up in near future. So I would be explaining my example, my this predictive tools by using some case studies, which I have developed for developing an anti-cancer compound for the treatment of ovarian cancer. So the two case studies will showcase two, because of time constraints, I could concise myself to two tools. One is how we can leverage the toxicokinetic data and use this toxicokinetic data for deciding the dose and dosing regimen for the phase 0 clinical trial. I would come back on by saying like phase 0 clinical trial. So I'll give you a heads up of that. What is phase 0 clinical trial? I would also like to show that how PBPK approaches can be used to understand the tumor exposure, and thereby we can provide, and how I provided guidelines for physicians for the dosing frequency and the visit frequency based on the pharmacodynamic response. So let's go with the first tool, which is utilizing the toxicokinetic data for dose selection and dosing regimen for the phase 0 clinical trial. So we know the normal phases of trials, like preclinical, then phase 1, phase 2, phase 3. So the phase 1 is more of a multiple ascending dose paradigm, but when we are very uncertain of a very new drug in the market at early stages, so we are very much at a risk stage of throwing this drug into phase 1 clinical trial, which needs a lot of formulation aspects, in which a high amount of resources are required for developing formulation, a large number of patients, and high doses, and a high risk factor as well. So recently, FDA has introduced an exploratory IND approach, or also called as phase 0 clinical trial, or also called as a micro dosing. So let me come to my, I'll come back on this part in my next slide. So the drug which we investigated is the heteroretinoid acid HA2. It has a good growth inhibitory range profile, which we tested in around 60 National Cancer of Institute cell lines. It shows like no toxicity in normal and benign cells and good tumor regression activity. And importantly, it was very safe. It has a very high no absorbed adverse effect level in rats and dogs, and lacks different AMEs and different mutagenicity types. It basically acts on the cancer cells by inducing apoptosis. Okay, so coming back to the phase 0 clinical trial, the main motive of the phase 0 clinical trial is to see what would be the pharmacokinetics or the dynamics with the first exposure in the patients. The main intention is to not have any therapeutic effect. Okay, the main intention is to see what would be the human exposure with a very limited dose, or a very limited dose, like let's say 1/50th of the rat NOEL. And at what dose or what concentrations can we achieve the drug concentration in the tissues which modulates the biomarkers level. So that's the main focus of the phase 0 approach, which FDA has recently launched, which is also called as an exploratory IND approach. So all these approaches, FDA is very wary of this fact that we need to develop new approaches and decision criteria for enabling the drug into the market and to the patients at very early stage with more confidence and less uncertainty. So we have to stay updated with all these approaches. So what approach I utilized is that my main aim was to get a first in human dose, and what would be the clinical trial regimen for the patients based on the toxicokinetic data. So I applied the concept of allometric scaling. It is in a very simple concept. It is extrapolation of the parameters like clearance, volume of distribution, distributional clearance, or absorption rate constants for a drug in a small species across different species. So we know these pharmacokinetic parameters from small species we can extrapolate to humans. So that's the concept of allometric scaling. It is also called as interspecies scaling. So don't get confused by two different terms. So this is a very important example which I would like to showcase to the audience, that how we can use the toxicokinetic data. But so so far the concept is like you use the toxicokinetic studies to make sure that okay, the LD50 is reasonable, thus it is safe, and we can go ahead with the dose of NOEL. But there is a lot of information hidden within these important data sets. So here, what we did, we got the data from our available resources for different species, and the solid and the dots and open and triangle, all these dots are the observed data. So what we did, we tried to characterize this data by modeling and simulation approaches. I would say right now modeling approach. So there's a difference between modeling and simulation. So modeling is basically you try to fit the data fit the data by using different compartmental approaches and estimate the parameters. And simulation is different. In simulation, you fix those parameters and try to make different scenarios. I'll show you those simulations as well, but this is more modeling. So we have to be very cautious by using both the tools together, when and where. So what we see, I won't go into details of that considering the time with what I have, but I would like to say that we see that there was a good concordance with the model prediction with the observed data for mice, rat, and dog. You see a different model for dog, because in dogs, what we saw that there was a delayed response as compared to mice and rat, and there was a prolonged and delayed response, which shows that there was a flip-flop kinetics in which the rate of absorption basically depends on the rate of elimination. So that's why I used a transit model to explain that delay. So that's a different direction I'm going, but just a brief I thought to discuss with everyone. So what we did here, you see different disposition parameters like clearance, distributional clearance, volume of distribution, and central volume of distribution and the peripheral volume of distribution. Okay, and on the y-axis you see the body weight, and on the x-axis different disposition parameters, the PK parameters. So from the model, as I said, that we estimate the PK parameters. So all these PK parameters we estimated for different species. And then what we did to get the information about human PK parameters, we scaled those PK parameters for different species across different body weights and came up with the human predicted PK parameter for clearance, distribution, which is intercompartmental distribution, the central and the peripheral volume. And we also wanted to make sure that this clearance is basically systemic clearance, or it is basically governed by different clearances, which comes mostly from liver or from kidney clearance. So we use some kind of a machine learning tool over here from GastroPlus, in which we used some QSR models, which were predetermined in the model to capture that this drug, based on its structure, what would be the main proportion of the drug clearance. So the white fraction, what you see over here, shows that the majority of the drug was cleared systemically from the body through plasma clearance by degradation by certain esterases and amides in the circulating in the blood. But very minimum proportion of the drug were metabolized by CYP enzymes in liver and kidney. So the majority of the clearance what we see is coming from the plasma clearance, and this is consistent across different species. That's what I wish to show using this machine learning approach. So what next? So the important thing is the more conventional way of deciding a dose is that you go with the highest no observed effect level, which could be the highest dose tolerated by the rat species. So in that case, based on the NCI data, we were having like 1500 mg per kg as the highest dose. So you go mostly 1/50th of that, but 1/50th of that as a first in human dose. But we need to dig into those information very carefully, because developing a formulation for the 1/50th of that, which means like around 30 milligram per kg, it will need a lot of formulation resources for developing a tablet or capsule for a 85 kilogram patient. So we really need to be very smart to decide like whether this is a real NOEL or not. So what we did from the modeling approach over here, we calculate the relative bioavailability. Like it means like what is the extent the drug is available in the system. So what we found that across different doses and different species, the extent to which the drug was available in the body didn't increases above a certain level at a certain dose, beyond certain dose. So that level was captured from this analysis, and that was more realistic bioavailability of the drug. So we used that dose as a more reasonable NOEL, and then calculated the first in human dose for the humans, which is like more reasonable, 2 mg per kg. So just think that you keep on adding drug to the system of animals, and the drug eliminates from the urine or the feces, and it do not reaches the circulation. So no matter how much you increase the drug, it might not reach the circulation. So even if it is safe, you will get a very higher starting dose for the patients. So based on our calculations for an 85 kg patient, it came out to be like 170 milligram. Any questions? I see raised hands. Okay. So what we then moved forward that, okay, how we can predict in humans. So we started by 2 mg per kg, and the plan was to move from 2 mg to 10 mg per kg. And on this plot, you see a human PK prediction. So the black line is the mean production, and always 100 patients might not have the same PK. So we add an uncertainty in the PK profiles by incorporating the 30% individual variability on the PK parameters for different patients. So always there is an uncertainty, so we need to capture that information that two persons might not have the same PK, but we can, while developing the the developmental plan for medicines, we need to always provide an uncertainty in the profile for the patient. So we decided, okay, we can start from 2 mg per kg and then we can reach till 10 mg per kg to cause target modulation, which is cyclin D1. Which reaches at 1600 microgram, which reaches over here on like 1000 nanogram per ml. So the next case study is, so Nitesh, how I am doing on the time? You can tell me if that's fine.
You have time, sir. You can proceed. I think
okay, so. Okay. Thanks, Nitesh. Okay. So the next approach I wish to discuss and share is that once we have the dose, we need to make sure that we need to know that when we should request the patients to come for clinical trial for autobiopsies, because these are ovarian cancer patients. They volunteered to provide their fallopian tube tissues. So we need to be very cautious and very careful in inviting in requesting the patients to come for autobiopsies, because those are very precious samples. So, Nitesh, can you ask everyone to mute themselves so that there is no interference?
Please mute all the participants. Only Ankush Sharma sir will speak.
Yes, sir. Yes, sir. I have done this.
Okay. Thanks, Urvashi. So, this is a very important tool just to understand like what would be the tissue exposures in different tissue organs. So what we did, like we used immunogenic mice, which are newt mice. We injected the tumor-bearing cells; those were SCOV3 cells, and then we injected in them the dye. So once the tumor sufficiently grows in these animals, they start to emit some radiation. So we captured those radiations to make sure that tumor is very well developed in the animal species, and this tumor microenvironment mimics the ovarian cancer patients. So basically, ovarian cancer patients, they have a very bulgy stomach because of the ascites, because of the fluid accumulation in their stomach, and they have a very critical situation. So we try to mimic that kind of condition in animals, and then we distributed the animals in different time points in a very staggered way, because those are very ill patients, ill subjects. So we have to be very careful in deciding the time points. And then we we administered our drug to those patients. And then, because this is more not an efficacy study, this is more pharmacokinetic study to check the tissue distribution or biodistribution, we collected the time point till 36 hours and collected the tumors and tissues and analyzed those tissues. So we, of course, we analyze the tissues by HPLC. So I didn't want to go in that direction. It's a very commonly used approach. But what I wish to share with you that once we have the data from different tissues, we have to characterize that data from a tool which is called physiologically based pharmacokinetic mode. So this tool is basically an arrangement of different tissues in a more anatomical way, but they are arranged mathematically, and in which each tissue is provided a supply of blood flow, venous blood flow, and arterial blood flow, and then from these tissues comes the venous blood flow and enters into the lung. So this is a very physiology of the entire body, and the drug movement within each tissue is governed by a fixed law of diffusion, which is governed by the permeability surface area and the concentration gradient. And this is basically the rate of the drug change in each of these tissues. So So it looks a very simplified, a bit of complicated model, but it has a very complicated set of differential equations to address the rate of change of drug in each different compartment. But I would like to provide you insight, like what was the main tissue of interest. Our tissue of interest was tumor, because we are developing an anti-cancer drug for ovarian cancer. So
Dr. Sharma, can we conclude the session, because now we have next session of Dr. Mahan Singh Rathod sir. So
okay.
after concluding this session, I requesting you on behalf of our PhD research scholar, because they all are doing PhD from Oriental or somewhere else, but they are still confused what they have to choose as a topic of research, because nowadays everybody is choosing plant for their research. So I think so you can guide them, that will be beneficial for them. So
Okay.
how it can be a socio-economic impact, because everybody is doing PhD just that they have different sort of purpose. But we want that if we will do PhD, if we will select research, just that can be helpful for future, for nation or world. So that will be more beneficial for all of us. So if you can enlighten our PhD research scholar in that way, so that will be very useful for all of us. Please, sir.
Yeah, sure. So it depends on if you are doing into herbal research technology. That's what I wish to like. I had a discussion with Dr. Nitesh as well prior to this presentation. So that's what I was trying to showcase over here. This is most of the concluding slides here, so you'll get an idea, and I'll share my thoughts as well.
Sure,
So
sure.
if you're using an herbal drug which has certain kind of let's say for example you're using some kind of chemo preventive or anti-cancer drug, right? So you can use cell lines. I believe you're working on cell lines, right? So so that's what I'm showing you here. So this drug is also derived from a natural source, which is a vitamin A analog or steroid analog. So you can do some Western blotting and get a sense of the dose response of this drug and different doses. You can get a sense of like what time course, so you can get an idea, and then develop some pharmacodynamic response for your drugs. So so that will give you an idea of what would be the dynamics going in the cancer cells, or for example, or you're doing a hepatoprotective activity or hepatotoxic effect or something like that, or a cardiovascular protective activity. So I'm just showing you here the cancer, but you can do this kind of studies for any type of and any type of pathologic condition. It could be cardiovascular, it could be hepatoprotective, it could be neurological. So each of them are designed from different cells, right? So you can do some cell viability study and then develop some kind of PD profile. You can before that you can test the pharmacokinetics of that compound, at how these compounds are behaving in different let's say for example rats. And then you can link those the pharmacokinetics with the PD part, and there are available tools like I would like to say how you can characterize. So this is the data generation part, right? How you can characterize those pieces of information. So as I mentioned, I don't know if you paid attention to that, but as I mentioned, there are available tools like Adapt5, which is a free tool from University of Berkeley. So from University of California, sorry. So you can use this tool for your model generation. So there are different trainings to develop a mathematical model to characterize the PK and the PD of the model, right? And then use some indirect response models to generate a response curve. So let's say, for example, your drug is kind of kills some tumor cells, right? So it will decrease, it will show something like this trend. And then you can use the pharmacodynamic concepts and parameters to get a sense of how the rate of change of response can be studied by studying the slopes of these curves. So but yeah, so that's a thing you can start with. If you have more questions, you can reach offline as well. I want to be cautious of Dr. Rathod sir time. so so that's how you can do. You can characterize the PK and PD data, and then come up with some strategies that okay, how would be the prediction. So why do we do this actually? Why I'm telling you to combine the PK and the PD together? Why? Because your main challenge is to see how the efficacy would be doing, right? That's why you're doing drug development, so that the patients get the efficacious dose, right? So you can plot both the pharmacokinetics and the dynamics over the course of time, and then see when you're getting the saturation in your dynamics. Suppose neurogeneration, for example, Alzheimer's, when you're getting a very steady state of the neurons in your brain, or when you're in, let's say for example, anti-diabetes formulations, when you see a very steady state in your glucose in the animals. So that is a point you have to select, and then suggest in your drug development protocols that this dose should be given regularly to the patients if we are going into clinical trials, and to maintain a steady state of the drug levels, right? So that's how I think there is a lot of training required for that to go till this level. But at least I thought to share an insight, a very high level insight, that how the young PhD scholars can think moving ahead in their PhD program. So you always wish to see not only the steady state in the pharmacokinetics, but also you have to see the steady state in the pharmacodynamics to have a better efficacious response. So yeah, with that, and this is the take-home message. And if you have any questions, you can shoot up questions offline or to my email. I'll try my best to answer your questions.
Yeah, Urvashi ma'am, now you can continue. If anybody have questions, you can ask. Within 5 minutes, we can have a question answer session. So from the audience, if anybody have questions from Dr. Ankush Sharma, so he can give better answers to you.
Yeah, feel free. This is an opportunity. Never leave it.
Requesting PhD is such a scholar because they have many doubt how they can choose their topic, how they can implement research for future socio-economic impact. Because research should be passionate, as well as that research should have some specific target also. So I think if anybody have some questions, this is a platform you can ask to Dr. Sharma. He's a brilliant mind. Actually, he was my batchmate, so we know him personally. So he will be a better person to answer all regarding actual research, pure research. So if anybody have some questions, so please.
Hello, sir.
Yeah.
Sir, I am Vivek Kasati.
Mm-hmm.
Hello.
Hi, Vivek. Hi, Vivek. I can hear you.
Yeah.
Yeah. Well, actually, I have seen that when we do this targeted study, as in my study, I have taken PIM kinase.
Mm-hmm.
So when I develop my drug, and actually I have synthesized my drug, and when I go through this kinase activity, then it is very, very costly, and it is not easily can be performed. So can you suggest how can we do in our this scholar line, that we can easily do these type of activities?
So you are limited by the cost of the kinase assay kits.
Yeah.
So I know that's I know your thought moving forward is very good, like appreciable, like you're thinking in that direction. But if it is a resource constraint, I understand that these resource constraints are really challenging. especially in India, and if we are really doing that high level of cancer biology. Um, but what we can do that always when we do cell viability assays, we try to at least have two kits. So that's what like we also did the cell biology essays and cell viability essays in my PhD program, and we were restricted by our grants, right? The PIs were restricted by the grants, so we have to use our kits very rationally. So I think the management for your college has to take care, at least like provide two kits, and as a scientist, as an early scientist, you have to do your experiments like very cautiously. So the first kit always goes in troubleshooting. You have to find what should be the right dose, what is the signal threshold you are getting in your, like this is some kind of like Western blotting. But I understand you're doing a tyrosine kinase activity, like, so you have to get a sense of what would be the cell viability at different doses, and once you get a and in that you have to make sure using a literature search that what is the concentrations in tumors of that tyrosine kinase inhibitor. Okay, so let's say for example your tyrosine kinase activity IC50 is certain X amount, right? X nanomolar or picomolar or something. So we always wish to see that at what dose we are getting like at what dose we are getting that concentration in our 96 well plates. Okay, and then see what is the response we are getting in the cell viability, and then use that concentration, right? And concentration, I mean to say the concentration which corresponds to your tissue concentrations in humans, like IC50 value or IC90 value. Those concentrations do a time response curve, and both of these can be done on single plate, right? So you have to design your experiments. That is very important. Designing experiments more rationally. That's what we learn in PhD. And if you fail, don't worry, you learn from that. If you don't fail, you're going to be learning nothing. So at least two kits will be sufficient for you, in my opinion. I don't know like what different tyrosine kinases you are using, but if you're using one drug, one kit for um, troubleshooting to get a sense like what dose gives what response, just to check the LLOQ, the lower limit of quantification of those responses, and then see what concentration we can move forward in my next essay, which corresponds to the tissue concentration in the human. So it will be very helpful when you will be publishing your information, because the reviewers will always ask you what was the decision criteria for moving forward with the time course action. Okay, so it has to be like the storyline has to be there, the justification has to be there. But like I would be restricted to answer your resource constraint, but I can tell you like within limited resources how you can plan your experiments.
Okay. Thank you, sir.
Yeah.
Neelanchal Trivedi, you can unmute yourself and ask questions. Neelanchal Trivedi, are you there? Okay. Now, Dr. Sunil Singh having question. You unmute yourself.
Yeah, good morning to all. Hello.
Good morning.
May I audible, sir?
Yeah, I can hear you.
Yeah. So my question is simple, because right now we are suffering from the COVID pandemic. So any anti-cancerous drug can be used as a treatment, as a pre-symptomatic treatment or treatment for the COVID-19.
You have to do research, because this work I presented in 2017, right?
Okay.
So I think and there should be, if you understand the pharmacology of the cell cycle arrest, there are definitely life cycle management drugs which are available, which can be tried to see how they affect the viral entry. There are certain drugs which also inhibit the enzyme which promotes the entry of the virus into the viable cells. So I know the mechanism wise, but I don't remember the drug wise. So, but the mechanism, you have to understand the mechanism. So how I would go, that I would learn different mechanisms, and the mechanisms of viral entry, or the mechanisms to re-engineer the viral re-entry. So they are different approaches, and then see different cancer agents who which work in those biochemistries, and then you can translate those anti-cancer mechanisms to see how the viral mechanisms can be modulated in COVID, SARS COVID viruses. So if I know the answer, I would have told Donald Trump, right?
Priya Dwivedi, you raised your hand. You unmute yourself and ask. Priya Dwivedi. Sheetal. Sheetal Sarsat. Are you there? You raised your hand. Okay, sir. No issue. Might be a mistake.
Okay, now I request Urvashi Sharma ma'am to conclude the session.
Thank you so much, sir, for sharing your valuable insights and knowledge on role of predicting modeling in pharmaceutical discovery with us. We are very much thankful to you for sharing your valuable knowledge with us. Over to you, sir.
Thanks, everyone. Thanks for insightful questions, and thanks for coordinating this session, and Dr. Nitesh, and the leadership team and the management for providing this opportunity to discuss the learnings and various examples. Hopefully, it makes some sense. Like if you take some 20% or 10% also, it would be really interesting and insightful for moving forward in your graduate programs. I would say in a PhD programs. So yeah, if you have any questions, you can direct those questions through Dr. Nitesh, and he can shoot up the questions back to me, and I'll try my best to answer if any questions have remained unanswered. So yeah, thank you very much, and yeah, all the best to everyone, and hope you my best wishes, and hope you succeed in your undergraduation, graduation, and PhD programs.
So once again, Dr. Sharma, really a very wonderful session. We have received many comments on chat box. They have enjoyed your session. Hopefully, they will learn something from you, and they will implement some concepts, whatever you have discussed in today's scientific session. So I think so. Now we will start second session. So Dr. Sharma now is in US. So I think there is a night. So if you want to stay with us, Dr. Sharma is most welcome. If you want to attend the session of our Honorable Dr. Mahendra sir. So
yeah, sure.
so,
Yeah,
yeah.
I'll as a, he's my honorable teacher, so I'll try my best to stay with him and hear from him and learn more from him. And thanks for consideration, Nitesh.
Thank you,
very much.
Ankur.
Thank you, everyone. Thank
Thank
you.
you so much, sir. Next, we have another renowned personality as a speaker, Professor Dr. Mahendra Singh Rathod, sir. Before moving further, I would request Dr. Mahavir Chajar, sir, Principal, OCPR, Oriental University, Indore, to please welcome, sir. So please unmute yourself. Dr. Mahavir Chajar, sir.
Am I audible now?
Yes, sir.
Yes, sir.
Welcome, sir. Welcome to our conference. And I hope you enlighten our participants with your expertise. Once again, welcome to. Welcome, sir.
I will try my best. Yeah.
Thank you, sir.
Thank you, sir.
Now,
Thank you
I
for
would
that.
I would like to introduce our next eminent speaker, Dr. Mahendra Singh Rathod, sir. Dr. Mahendra Singh Rathod, sir, is the Director at Internal Quality Assurance Cell, Maharishi Markandeshwari University, Haryana. Sir has over 15 years of experience in which he has worked at various esteemed positions in various reputed organizations such as ISF College of Pharmacy, CT Institute of Pharmaceutical Sciences, BRNCOP, etc. Sir has also worked as a member secretary of Institutional Ethics Committee registered with CDSCO, Government of India. Being an academician, Sir has completed various research projects, guided so many students, and organized conferences at various national and international levels. He has extensively published articles for several peer-reviewed journals, written books, and acquired patents by his name. We welcome you, sir, and it's a great honor for all of us to have you here. Requesting you to please take over the session.
Thank you, Ms. Yarushi, for the nice introduction.
Thank you.
And I would like to thank the organizers of this international conference. And firstly, I would like to start my session. Before that, I would like to quote that this is really a very fantastically managed international conference that so far I have seen. So it's just like that we are sitting front to front and talking to each other. So this is the congratulations to all of you. So I hope I am audible to everybody.
Yes, sir.
Yes, sir.
It's clear.
Yes.
So I will start the presentation by just let me share my screen with you. So is it visible?
Yes.
So before moving with this topic that is relevant, so all of you might have heard about these recent changes, amendments in the clinical trial rules, that is Schedule Y of the Drugs and Cosmetic Act 1940. This is very well renowned in our country and is very much popular among the person who are working in the clinical research. So this is a recent change. So that I thought this topic was selected particularly for the students who are attending the conference, and then particularly it would be useful when they will be going for outside their for searching the jobs, and this is a particular segment which is rising, which is on the boom. So I will start my presentation with this current perspectives of the new drug and clinical trial regulations in India. So everybody is talking that the clinical trial market is huge and there are a lot of opportunities available there. So let's talk with some data that why this is so. So we can see that there is a global contract research organization market size was estimated as US $34.5 billion in 2018, and it's projected to rise by 55.3 billion by 2024. And this cumulative accumulated growth rate is expected to have 8.2. So this is a tremendous growth rate, and that is being going to about to observe. Even in this pandemics, these clinical trials are not stopped; these are being continuously going on, so that you can see that whatsoever the adverse situations are there, this is the evergreen field for the choosing. So let's talk about the Indian clinical trial market. This market trial size is expected to reach up to US $3.15 billion by 2025, and is projected to register a growth rate of 8.7, and now it will be more. So what are the key factors about that? That is the globalization of the clinical trial, and then there is the adoption of the new technologies in the clinical research. Dr. Ankur has previously, in the previous lecture, he has shared that what is the preclinical development, and then after preclinical development, there would be a clinical phase in which clinical research principles would be applied. So then another factor is this growing the disease variation and prevalence. Along with that, the increasing R&D promote outsourcing. These were the key factors that were driving the clinical trial market in India. So we can see that why this India is considered as the global destination for the clinical research. Everybody is saying that the various multinational companies are coming to India for conducting the clinical trials, clinical research. So previously there were, I will just share with you some examples here later on, that what happened in the clinical trial industry in the mid of this decade. So clinical in India, why India is considered as the good destination for the companies, and why there are opportunities also for our students. So you can see that we are having the population nearly about 1.3 billion and having the various genetic background. And then this it is offering a large treatment native pool for the development of therapeutics on a range of disease conditions, and then most diverse disease profile due to the economic, environmental, and ecological variations in the 29 states of the India and 7 union territories of the country. So here is the rapidly transforming healthcare market is there, and highly educated physicians and cost competitiveness, and the most important factor that is attracting the global healthcare research organization or clinical research organization here, that the cost is around 40 to 70% less as compared to the Europe and US market. So we are saying that there are a lot of factors which are favoring our country to become the global destination. Then what are the what were the various factors due to which this there was some lag phase that was observed in the market of clinical trial in India in 2013 to 15? The reasons behind this were the red tapeism or governance, and along with that there were regulatory uncertainties. So before that introduction of this reforms in the clinical trial rules, what was previously the clinical trial was not that was regulated, and so forth. There were a lot of uncertainties were there, so that people or investors were hesitating to move here in this. And so suppose something happened in some clinical trial, then our so-called socialists they raise their voices. Foreign companies are coming here, they are doing clinical trial on us, and then this is for the benefit of their own population. So likewise, these questions were raised. So this was due to the red tapeism of the governance and the regulatory uncertainties. So one example was there that where one principal investigator was found to have generating the fraudulent data and referring the patients from a government hospital where he was working to his private clinic to gain more income. So these type of incidences and has attracted the attention of the Supreme Court of India, our honorable highest governing authority, to address this issue, and they have instructed the central government to reform about the clinical trial rules. So that's how this thing were emerged, and there were emerging number of the there was emerging need for the clinical trial and clinical trial rules. So it was essential that all clinical trials in India should be conducted as per the International Conference of Harmonization Good Clinical Practices Guidelines. Along with that, these factors compel the government to bring the new drugs and clinical trial rules. So I talked about the opportunities in this sector, in this segment. See, for students, I would like to say that these are the sectors, you can say the clinical research, and another one is the part of the clinical research is the pharmacovigilance or post-marketing surveillance. These are the two highest growing sectors. You may see the Naukri.com or any job portal, you will find that out of 10, 6, 7 are from the pharmacovigilance or clinical research jobs. So why this opportunity is there? Because there are a lot of industries or R&D organizations which are investing in the clinical research organization or contract research organizations, and there is an increased demand of the trained professionals. And so the understanding by this background, just I would like to say that understanding of these new clinical trial rules, new drug and clinical trial rules, is imperative for every pharmacy professional to understand what is this field is, and to unlock the opportunities in the clinical trial industry, those who are interested. So here, this is the official Gazette of India that has shown that on 19th of March, these 2019, last year, these clinical trial rules were in force. So here there are 13 chapters in this clinical trial rules, and all were effective from 19th March, except this chapter 4 that is related to the ethics committee regulations. this was coming into force after 6 months or 180 days, that is from the 21st of September 2019. So the brief about these rules are having the 13 chapters, that is including 107 rules. There are 8 schedules, and these new rules will apply to all new drugs, investigational new drugs for the human use, clinical trials, bioequivalence and bioavailability studies, and ethics committee. The new rules will supersede the Part 10A of the previous Schedule Y of the Drugs and Cosmetic Act 1945 and go into effect immediately. And this drug is for the, however, new drugs and clinical trial rules are applicable for the new drugs or investigational new drugs. And the veterinary drugs which were being under the clinical research veterinary use, they were following the earlier regulations. This rule would not be applicable to them. So this particular document that is released by the government of India is a very huge document. It's around 160 pages. So it's not possible to cover each and every aspect of this new drugs and clinical trial rules 2019 here. So, but however, I will try to cover in brief and highlighting the salient feature of this rules. So I would say in the beginning that these new regulations are welcome step by the government of India. And these are aimed at promoting the clinical research in the country by implementing time-bound review of the application. Then they're facilitating the indigenous research. They are allowing the increased predictability, the transparency of the regulatory pathway, and providing the clarity on many complex subjects which were not available previously. So here we can see this definition of the new drug. So definition of the new drugs is a very important aspect of this new clinical trial, new drug and clinical trial rule. You see, this name of this rules are new drug and clinical trial rule. So new drug is very important aspect, and we have to understand what is the meaning of the new drug. So according to the definition, this is including active pharmaceutical ingredient or phytopharmaceuticals that have been used in the country to any significant extent, except in accordance with the provision. And these drugs should not be have been approved as safe or efficacious by the central licensing authority with respect to its claim in any country. Then there is a second point, that is the drug which is approved. This would also considered as a new drug, that is a drug which is approved by the central licensing authority for certain claims and proposed to be marketed with the modified or new claims, including indications, route of administration, doses and dosage forms. So basically you can say nowadays in COVID-19 pandemic, we are saying that hydroxychloroquine and many drugs have been repurposed for treating this. So these would be coming considered as a new drug on the expedite pathway of the approval, and this would be coming into this category. So they would be considered as a new drug because the drug has been approved for the different indication, but now it is going to be used for the different indication. So it would be considered as a new drug. Similarly, the fixed dose combination of the 2 or more drugs. So this is a very important thing that we have to understand, that the 2 drugs which have been approved previously separately for the different indications, and there is a combination of the 2 previously approved drugs in a fixed ratio. If somebody want to have approval or product registrations, then he has to conduct the clinical trials for considering these new drug rules. So this is including the indication route of, if the route of administration or doses or dosage form is being changed, this would be considered as a new drug. So these first three categories would be have to be looked into, and the data is to be submitted to the government for 4 years. It would be considered as a new drug, and there is an opportunity lying behind these things. I would just finish this thing, and then I will discuss about this opportunity. So the fourth point is the modified or sustained release forms of a drug or novel drug release system, that of any drug which is approved by the central licensing authority. Say for example, any novel drug release system, liposomal formulation, or sustained release formulation of any existing drug has been developed. This would also be considered as a new drug. Then the vaccine recombinant deoxyribonucleic acid and this biosimilars, or you can say the monoclonal antibodies, stem cell derived products, all these would be considered as a new drug. This fourth and fifth point would be considered as a new drug forever. These would remain forever as a new drug, and then regulatory authorities require the submission of the documents for this. I talked about the opportunities behind this. See, you can see, in beginning, I also shared that the clinical research and the pharmacovigilance is carrying a lot of opportunities for the budding pharmacy professionals. The reason behind that is there, because by laws it was mentioned there that to monitor the pharmacovigilance, all of you are understanding what is the meaning of pharmacovigilance and post-marketing surveillance. So this is just to know about this adverse drug reaction reporting after the drug is approved in the and it is in the market at the larger scale. So the regulatory agencies are interested in knowing for these first three categories, it would be for the 4 years, and for the fourth and fifth category, it would be for the 4 hours. So the post-marketing surveillance data is to be submitted to the regulatory agencies. So you see this how huge opportunity is lying here. So this post-marketing surveillance and the other serious adverse event, if any occur with the drug, has to be sought after by the regulatory agencies. So we can think about this. This new drug is a very important definition. That's why I took a lot of time here. Then there is a clinical trial definition. Earlier, these definitions were not being very much clear. So now everything is clear that any study, systematic study, whether for the development of the data or discovering or verifying its clinical or pharmacological, including pharmacodynamics, kinetics, or adverse effects. You see that here the adverse effect has been mentioned here. With the objective of determining the safety, efficacy, or tolerance of such a new drug or investigational new drug. So in this new drugs and clinical trial rules, there is one more new term has been introduced, that is the academic clinical trial. So academic clinical trial of a drug that is already approved and initiated by any investigator or academic or research institution for a new indication or new route of administration or new dose or new dosage form. This is very much relevant to the people who are working in the medical college or the PhD students who are working on the PhD projects, that if they want to do something in this sector. So in the red ink, I have mentioned that these type of studies should be intended to be used only for the academic or research purpose. And it should not be used for the seeking approval of the any regulatory agency or licensing authority in any country for the marketing or commercial purpose. Then this trial would be known as the academic clinical trial, and it would be having certain exemptions that these type of clinical trials, which are for the pure research purpose, need not to be the permission need not to be taken from the DCGI, Government of India. Only the ethics committee would be enough to give the approval for these. Academic clinical trial is having some salient features, that is for the approved drug. I already covered. Then this clinical trial initiated this for the academic or clinical research institutes. So these are the certain features for the academic clinical trial. This is a new term. Then there is another aspect, that is the regulation on the biomedical and health research. In this new rules 2019, this research has been defined as including the studies on the basic, applied, or operational research or clinical research designed for primarily to increase the scientific knowledge of the disease or conditions. So these biomedical and health research was earlier being guided by the ICMR guideline. ICMR's guideline has given ethical guidelines for the clinical trial or the drugs or you can say the studies which are being conducted on the human beings were guided by the ICMR guidelines 2017. These are guidelines; they were not rules. So there was a regulatory uncertainty, and it was creating very much problem. But now it has been covered under the rules, so that this becomes law. So in this basic regular biomedical and health research covers the in vitro diagnostics, performance testing for the research, new surgical intervention, public health survey, epidemiological health survey, observational and non-interventional studies of the old drugs. The compensation and calculation guidelines are not, these are the things that would be covered under the biomedical health research. And now these would be covered under the rules. However, the compensation is not made clear here for these. If something happens in the academic clinical trial, then what would happen? What would be the compensation guidelines? Because in the professional clinical trials conducted by the investors, investigators, or the sponsors, that would be very well regulated. And I will share the example of this compensation calculation at the end of the slide. But in this case, it is not, still this is a gray area which need to be improved. We all know that if anybody wants to conduct a clinical trial in India, then firstly he has to take the permission from the Drug Controller General in DCGI. Then there should be approval taken from the respective ethics committee where the study is planned. And then there is a mandatory registration on the Icipmbs maintained website, that is ctri.in. So these are for the professional clinical trials which are intended to generate the data for the product registration in India. This is not applicable for the academic clinical trial, as I previously shared with you. So here in this rules, particularly the ethics committee has been given a lot of significance. So since I already had served as a member secretary for one of the ethics committee, and this is, and I am having experience that getting approval of the ethics committee by the Central Drug Research Standard Control Organization, CDSCO, is very difficult. And what the questions they are asking, that I cannot share that everything they are asking, they are very particular that what is the thing, how it will be happening. So there are certain guidelines, there are certain changes occurred here. That is, there should be minimum 7-member committee should be there from the medical, non-medical, scientific or non-scientific areas of interest. That it should consist at least one lay person who will represent the society, then one woman member, then one legal expert, one independent member from any other related field, that is such as social scientist or representative of any NGO or philosopher or ethicist or theologist. So this is the composition. Minimum these 4 type of people should be there, and then of course the clinicians should be there. Along with that, one member secretary would be there, one president would be there, chairman of the committee would be from the outside of the host institution. So these are the things which were covered under these rules for the ethics committee. Then there's this is the has been change made here, that at least 50% of the members who are not affiliated with the institute or organization in which the EC is constituted. So there should be 50% outside members, so that this steps has been taken to increase the transparency of the decisions being taken in the clinical trial approval protocols. So every members of the ethics committee shall be required to undergo the training on the recent good clinical practices, GCP guidelines, ICH guidelines, and this regulatory agencies is asking a certificate for any member that what is the qualification, how trained he is for the recent trials. So these are the things we have to submit to the regulatory agencies. So what would be the impact of these changes here in the ethics committee? That the institutions may have the several ethics committee, because I told you that there should be 2 ethics committees have been mentioned. These need to be constituted. One is for the academic clinical trials, and the second one is for the clinical trials and the bioavailability or bioequivalence studies. The validity period has been increased from 3 years to 5 years in these rules, and there would be the renewal of the registration of ethics committee 90 days prior to the expiration of the current approval. Then if any changes there in the membership or the constitution to be intimated to the DCGI within 30 days, so that the DCGI is having very close check, because this is a very important committee because DCGI cannot look after the many clinical trial or many clinical research in the country, so that ethics committee is empowered to monitor and to give the approval. On the one hand, on the other hand, the responsibilities of the ethics committee also increased by virtue of these laws. Then there should be the records of the ethics committee or the proceedings of the ethics committee are to be maintained for 5 years. This is mandatory by these laws. If any suspension or cancellation, if any fraudulency or any shortcoming is being found in the functioning of the ethics committee, then show cause notice, warning letter, or rejection, or suspension, or debarment are the steps that can be taken by the regulatory agency, that is CDSCO. Then there should be a separate ethics committee to be constituted for clinical trials or BA/BE studies and for the academic research. I already narrated this thing. Then there should be the application for the permission to conduct the clinical trial of a new drug or investigation new drug on the form CT04. Earlier, this was the form 44 in the previous Schedule Y of the D&C Act. The application fee also has been changed. So I will share these new drugs and clinical trial rules with the organizers and this, of course along with this PPT, so that many students can be benefited. If anybody want to study further about these rules, can refer these documents. Then the grant of the permission. So in this clinical trial rules, the process has been made expedited. Here, the grant of the permission to conduct the clinical trial, the decision would be taken within 90 working days. And here one more automatic approval process has been given for the. This is for the 90 days is for this all other countries or multinational companies. While if the clinical trial is indigenous, that is rising from India, there is a provision of the automatic approval. That means if the products which are being developed indigenously, the applications to conduct the clinical trial will be considered approved if there are no queries raised by DCGI within 30 working days of application. So that if indigenous clinical trial is somebody want to get the product registered in house or in our country, and they want to conduct the clinical trial, just they have to apply on this CT04 and wait for 30 days. If any comment is coming there, then have to reply. Else, it would be considered automatically that your application is approved, and you can start the clinical trial just by sending an intimation letter in the form CT4A to the DCGI. So this is a welcome step by the Indian industry or the Indian researchers who want to develop the indigenous products. Now moving ahead with this, there are various conditions of the permission for the conduct of the clinical trials. So here you can see that previously this thing was not clear. Now it is made clear. Suppose an institution or a researcher want to conduct the clinical trial, and the ethics committee is not available with their institution. So what they can do? They can go and search nearby in the periphery of the 50 kilometers within the same city or within the radius of 50 kilometers. If any other institute is having the ethics committee, they can get their protocol approved here and they can conduct the clinical research at their own institution. So this is the thing that has been given in these rules. Then the status of the enrollment of the trial subjects shall be submitted on the quarterly basis. There's a 6 monthly status report is being asked by the DCGI. Along with that, the termination of the clinical, if the clinical trial is being ceased or terminated, then the notification should be sent to the DCGI within 30 days. The validity period of the permission to initiate the clinical trial is within the 2 years of permission. If the permission has been taken from the DCGI, the researchers have to start the clinical trial within 2 years of the permission. Then there are provision of the post-trial access of investigation new drug on a new drug. Then academic clinical trial is there. I already narrated that there is no need of the definition this permission required for the conduct of the academic clinical trial. Then there is a provision of this one important thing that has been mentioned here regarding the post-marketing surveillance. That previously the incidents the serious adverse effect is to be intimated within 14 days to the DCGI or the regulatory authorities. Previously, it was the date of occurrence, but now it has been converted to the date of knowledge of occurrence of SAE. That's mean if it is being established that the particular death or particular injury to the patient or the participant of the study has been caused due to the particular drug itself. After establishment of this fact, within 14 days, the company or the organization has to report to the DCGI. Previously, it was the moment it is occurring, within 14 days you have to apply. So that lot of ambiguity was there. This pharmaceutical industry is used to prove that this has not been occurred due to the drug, particularly. There were another reasons for the death or for the injury. So this knowledge of occurrence of SAE is the important term here that has been introduced and made more clear. Then there are countries for which this approval, new drug approval can be given. The DCGI is having its own discretion for the approval. Then the order of suspension or revocation in the public domain, the DCGI can give this instructions. There are particularly specific requirements for the phytopharmaceuticals. Previously, this phytopharmaceuticals related law was silent about the clinical trial aspects, but now it has been introduced. Then PSUR content and structure is aligned with the European guidelines. So PSUR is the public safety updated report, periodic safety updated report. So here is these are the slight features of this clinical trial. But I believe that the students, particularly who are going outside to their program after passing the, let it be B Pharm or let it be Masters in Pharmacy or the Pharm B students, they should be at least going through these recent trials, because in the interview definitely these questions would be asked, that what are the recent amendments. So that this was my thinking behind choosing this particular subject, as it is new and it might be helpful for them. Now I told that there is a compensation aspect of this clinical trial, and there's a huge compensation is to be given if something happened wrong in this clinical trial. I have taken one example calculation for this thing for the clinical trial, which are intended to get the product registration. So the compensation would be calculated by the formula, that is B into F into R, where the B is the base amount, that is 8 lakh. It has not been changed previously. Previously also it was the same, and now it's also. Then there are the factors which are depending on the age of the trial subject. In Annexure 1, it has been given, and I have taken the example value. Suppose the person is of the 25 years, the value is coming 216.91 for this. So here in this document of the clinical trial official gadget, the things are being given, nicely mentioned there. The table is there. Then the R is the risk factor, that is depending on the seriousness and severity of the disease. That is the presence of comorbidity and duration. So there are, this is the point has been given 0.4 to 4 as under, that if 0.5 is there, if the patient is terminally ill, one score is to be given if the patient is at the high risk, 2 is for the moderate risk, 3 for the mild risk, and the 4 is for the healthy volunteer or trial subject. So the compensation calculation in the case of death, let's take an example. If the person of 25 years of age is dying due to the clinical trial, and who is not having any risk at the time of enrollment in the trial, then the compensation to his legal hire would be calculated like this. And this compensation amount comes around 69,61,940 rupees. So this is the compensation that has to be given in the case of death of the clinical trial. So these are the things that I like to share here. And here are the, I declare that nothing is there. Only this example has been framed by me. By the rest of the things I have taken from this internet and the other periodicals, the recent data I collected. These are the references here. So it's my presentation completed. Thank you very much. I welcome any question if it is there by the participants. I would be glad to answer that. And if anything related to the, since I am also teaching here in the Doctor of Pharmacy PharmD program here at my university, along with this my current portfolio as director of the IQAC. So I would be answering you better.
Thank you so much, sir, for a very wonderful session. And it was really a very informative session for all of you. So I request all the participants, research scholar, now platform is open for all. Sir can give answers to all. Yes. Yeah, please, anyone have question, they can ask questions directly to Dr. Mahen Singh Rathod, sir.
Participants, please behave yourself. Do not annotate. Okay, please maintain the decorum. Rathod sir, you can stop sharing.
How it would be?
Dr. Rohit here. Thanks, Mahendra, for giving that much of informative session. I'm not having any question, but just I would like to say you have covered very well, and I think you have just shown us the recent changes which DCGI has made it for clinical trial, and actually it was very bad shape in near about 2015 to 16, 17. So now we can hope the more opportunity will come into this industry, and the process will be very fine and very it would be very thorough, so we will get benefited. So thank you very much for taking this session on this topic, and I think it has been covered very well, what is the current requirement and what is the current changes. So as a clinical trial industry professional, we should know all these things. So thank you once again.
Thank you, Dr. Rohit.
Thank you. Very nice compliments are coming in the chat box also. So it's really informative session.
Dr. Mahen Singh Rathod, sir, actually, I have one question from you, sir.
Yeah, please.
When we were students in NATA, actually, you are doing one project, academia and industry collaboration. Now, everybody is taking this topic all over the globe, but still we are lacking to maintain this bridge in between academia and industry. So how we can fill this gap, sir? So we want your opinion and how we can improve this particular correlation in between academia and industry for the better outcome of research, sir.
Yeah, I would like to take a few minutes over this topic because it's very important and relevant. Basically, what is the root cause problem? Why this academia and industry interaction has not taken shape in our country as it is in the Western country? So the basic root cause is previously it was considered as a syllabus that we are teaching in our universities or colleges. Then it comes about the resource settings, that we always complain about the poor resource setting here, so that our clinical trial is not being our research is not up to mark or up to date according to the industrial standards. Industry do not take interest in our researches. So this particular project was taken with the aim to bridge the gap and to maintain or develop a database in which whatever the academic researches we are conducting, these academic researches would be put together on one platform, and this would be shared with the industry. See, what was the basic purpose? I'm telling you that there should be a basic philosophy behind this. See, academic research is having the philosophy that whatever we are conducting research, the student is working for 1 year in a master's and 3 or 4 years in a PhD topic. If the results are good, then it's fine. It can be shared. Even if the result is not good, or you can say that its outcome is not clear, this particular project is useful for the degree awarding, isn't it? But industry cannot afford this thing. Industry cannot go for the vague ideas, or I would suggest the budding scientists, let's take any foolish idea, I will say, and work over it. It might be possible that there might be success, and you would be agree with me that all the great researches has been started with the foolishness. because it would be out of the box thinking should be there, because if you will thinking out of the box, then people will say कि दिमाग चल गया है इसका, ये अभी कुछ नहीं कर पाएगा, likewise इस तरह से बातें होती हैं.
True.
This is the matter of honesty, dedication, whatever we are lacking in, especially in academia, because academia persons are doing the research what they want just because to get the degree.
I'm coming over this point that we have to, this is one thing that we have to change our mindset. Yes. And the second thing is that we have to dare to think out of the box.
Yes, sir.
That's mean we are still sticking in the pharmaceutical industry about the R&D opportunities. The master student is thinking कि मुझे R&D में job मिल जाए तो मेरा life set हो जाएगा. आगे जो B.Pharm student है वो कहेगा कि मुझे अभी शुरुआत में 20-25 हज़ार की job मिल जाए, that would be, I would be set like this. So this is the mindset of the students. That the very narrow opportunities we are taking after doing our graduation or post-graduation. But nowadays you see the lot of the paradigm has been completely changed. See, this pandemic has changed this method of teaching. That from the traditional books now gone. You understand? You take this my words. This traditional book system has been gone. Now this would be the new paradigm that was taking shape. Along with this, there are a lot of the opportunities for this is particularly for the students who can take their career shape in these aspects. As I told you that clinical research is there, one more pharmacovigilance is there. Then there is one more thing is that is health economics and outcome research. That's a HEOR. See, the providing the affordable health is the priority of any nation's government. In India also, the government of India is also giving this. So this health economics and outcome research of pharmacoeconomics is one segment in which one student, one person can think about making the career. Along with this, for the people who are having expertise in the pharmacological or you can say clinical research aspects, they also can go for the new avenue, that is medical science liaison officer. This is the extension of the medical regulatory affairs officers in the industry. So this is one, you can search on the internet. It is a very huge topic. I can take one full lecture over it, but the time is not permitting.
Definitely, we'll organize very soon your expert lecture for our industry by online mode. Definitely, sir, on especially Indian industry collaborations.
Along with that, what the takeaway message for the students particularly, I would like to say that keep yourself updated about the recent changes. Now you see, I'm not complaining, but the thing is that previously 1 or 2 years back, there were the e-pharmacy regulations were drafted by Government of India. And then the comments were asked, how many of us are writing the comment to the Government of India that since it is concerning with us, this is concerning with our pharmacy professionals. So we should be aware that what are the regulations being going there, and these are going to govern our livelihood in coming future. So we should be remain updated, abreast with the latest knowledge. Number one thing. Number second thing, in the you can see that previously one law was there, one regulations was passed by the Government of India in which this medical devices, for that the drug inspector, where nowhere was the pharmacy qualification was mentioned here. You see what we did? This before making any law in our country, there are the time period is being given and is open in the public domain that anybody can give their suggestion, so that any amendment is required in the law that can be done. So this law was passed and there was no qualification of pharmacy. You see what was the qualification? The B.Tech graduate was eligible to become the drug inspector for the medical devices regulations. This is sole our right. We have studied 4 years. We have studied 2 years PhD. At least one person who is doing PhD is spending 10 to 12 years of his life, and our qualification is not there. Whose fault is this? That this is the thing that we have to. I'm talking that we have to think out of the box, and we have to raise our voice, because one single voice would make the huge noise if taken cumulatively. So this is my message to all. I hope that I have taken a lot of time beyond this assigned time.
No, it's okay, sir.
So this is all from my side. Anything else? Again, I will congratulate Dr. Nitesh to you for this such wonderful and of course, I have seen one thing I would like to comment. Actually, in the beginning also I was about to comment.
Yes,
that the teamwork is evident from your team.
Yes.
That everybody is working synchronously and without any. I have not seen any haphazard manner thing that usually occurs.
It's all the dedication, commitment, and devotion of my team, sir.
Definitely. You are having really very
all credit goes to our team, dynamic team, sir. Definitely, sir. They are working even in night. They are not thinking about where's the morning, night, and all. 24 hours are online, sir. So really, on behalf of you, I really congratulate all, sir.
So this appreciation from your side means a lot, sir.
Definitely, whatever I have observed that I have shared with you. So
thank you.
Congratulate to all of you, and I hope that in the future also you would be doing well and conducting such good things. So this is all about from me. This is over to the organizers, the master of the ceremony.
Thank you so much, sir, for such an elaborative and informative session on current perspective of new drugs and clinical trial regulations in India. We are glad to hear you. Thank you once again. Now, I would like to request Dr. Sumit Dwivedi, sir, to please felicitate Dr. Mahendra Singh Rathore, sir. Sumit, sir, over to you.
Hello. Is I audible?
Yes, sir.
Good morning, sir. It was a really very nice session. You have delivered and you have enlightened a very good approach of the pharmacist and drug development. We, on the behalf of Oriental family, thank you for your concern and enlightening us in this conference. Thank you so much, sir.
Thank you very much for inviting me.
Ma'am, now Dr. Piyush Jain sir is also here, I think.
Yes, sir.
On behalf of Oriental University Indore, really it's a matter of to welcome Dr. Piyush Jain sir.
Thank you. Thanks a lot for having me.
Thank you. So what we have to do, we will continue the session or we will take a break, ma'am? I think we can take a break, right?
Yeah, deserve a break, you know, after a long 2 days.
Okay. Yes. So what you want, sir? I think.
Are you asking to me?
Yes, sir.
No, definitely take a break, you know, so then everyone can listen this last keynote lecture with their full concentration.
Yeah, definitely. Because we can't offer online high tea, sir. Otherwise, they will enjoy a lot. So I think I request.
No. It's really a good idea because I have really a lot to talk about and probably the topic and the research you might have.
And then we are excited to listen to your scientific session, sir. So one thing, take a break. So Urvashi ma'am, just announce the break.
Okay, sir. So now here we will have a small break and we will resume with our third session at 12:15 p.m. Thank you so much for connecting with us and requesting you to join again. Thank you so much.
Thank you, Dr. Mahendra Singh Rathore, sir. Thank you so much, sir.
Nitesh, sir?
Dr. Nitesh, sorry to interrupt in between.
Nitesh, sir, are you there?
Excuse me.
Yes, sir.
Can I leave here the session because I have to attend some.
Yes, sir. Yes, sir. You can, sir. You can.
Thank you. Thank you very much.
Thank you, sir.
Congratulations.
Mahavir, sir, shall we end the meeting?
Hello, ma'am.
Yes, ma'am. We will resume at 12:15.
So, sir, shall I end the meeting then now?
Yeah, sure, ma'am. Sumit sir कुछ कह रहे थे.
Sumit sir?
Yes, ma'am. Actually, I think Nitesh sir is not there, so we'll end the meeting and we'll resume by 12:50.
Okay, so I announce event management committee to come on conference video call, please. Right now,